मला बघा थोडा वेळ तर.
Hello, hello. Okay. So where should I? Okay. Uh, should I move or no? Okay. Okay. No problem. Okay. Hello. Am I audible? Hello. Yes. So, dear students, today we are with the drug designing. Now, before coming to this, I just want to ask you few questions. I just want that first of all you tell me what do you know about the drug design, or Hindi, English, Marathi, everything is okay for you? Which is more preferable? English? Marathi पण चालेल? But I am not Marathi, okay? But my Marathi is not that okay, right? So you will laugh if I'll speak Marathi. So better to, I'll go with the mixed part, yes. So now, what do you know about the, anything about the drug design? What is drug? Anything you know about it? By this time, now all of you are in UG, right? So now you must be knowing what is drug. Chemical composition of certain chemicals, specific chemicals. Okay, fine. Now, suppose if a drug is to be discovered, okay, how much year it should take to go about it? How much years it takes? What do you think? 2 to 3 years? What about years? How many years? 2 to 3 years? All of you agree with it? Are you? You also agree? Yourself? You also agree? It takes 14 years to take. It is 14 years are required for a drug to make. So it is a very, very huge and long time span. And by that time, the new drug comes. That means the disease is, you are resistant to that disease. Okay. Earlier, when we were having fever, we were given, I think, 250 mg crocin. Okay. My dadaji and my dad and then myself, we were up to that only. But now when you go to maybe that as a protein. Okay. Now we have another target, biochemistry and biotechnology knowledge. So you might be knowing about the lock and key theory, correct? So now see, what's your name? Can you please stand up? If I give her, if I give Shraddha, Dhundo Chabi, has some specific alignment in the liver, you treat it as a receptor. Okay? Receptor, right? Now, there are some active site in the receptor. Okay? But now, this flexible. ये अंदर चेंज हो जाता है. चाबी काम आएगी? नहीं आएगी. अब मुझे करना क्या होगा? मैं तो फंस गई. ठीक है? Active site. Got the point? What is active site? That is the active site which is there in the liver. This protein is set, receptor is same. Okay? So that comes under SD, SBDDR based drug design. कभी चाबी बदल दो, कभी ताला बदल दो, एक्टिव साइट चेंज किया तो उससे फिर आपका ये बदल जाता है, राइट? देन यू हैव टू फाइंड आउट द एक्टिव साइट. एक्टिव साइट में कौन सा की बैठेगा, दैट की यू हैव टू प्रेडिक्ट एंड यू विल गो विथ इट. Okay, so that is one type of study which is called as receptor based drug design. And structure based drug design means मैंने श्रद्धा को चाबियां दे दी लेकिन सारी चाबी फ्लेक्सिबल है, ठीक है? अब आप एक रिजिड ले लो, उसमें से सारी चाबियां ट्राई करो, which active site will be fitting it, that will be your key. So that is very from this. Okay, न मेरे पास लॉक है न मेरे पास कीज है. So now that is called as de novo drug design. Now what I will do, okay, I will take one molecule. Okay, I'll say that, let us say for example, this is carbon, this is oxygen, this is hydrogen. Okay, COH bond, right? Now I'm telling you, remove this H, put Cl there, put another thing there. Okay, CH3 group, OCH3. I am up to this number. Are you getting my point? देखो, it is as simple as that. मुझे घर लोकेट क्या है मेरे पास and I'll make it. Okay, but जब मैं आपको बोलती हूँ कि आपको ये पोटैटो दिया जाता है, टोमेटो दिया जाता है और मटर यूज करना पड़े, ABDD, you are given structure and receptor. Sometimes receptor is flexible, so things are going vice versa. But now when you come to de novo, you have to have your own molecules. You can have your own library. So I prepared 50 compounds, suppose with one molecules you have prepared. This structure again, I don't have time. I hope you know what is in vitro and in silico. Okay, computer based BDD. Computer, this is our syllabus. In this we have RBDD and SBDD. This together is called as computational based. Got it? All of you? Any doubts? So now here we should say that, or screening you have to do now. Okay, so now in de novo you have prepared 500 molecules, and then you do the trial and error in computational site, wherein the receptor will have the active site. Flexible is the receptor. Okay, first thing. Suppose one thing becomes constant, one thing is fixed. Now if I hold this hydrogen or this OH group below, that have this, and now there are conformers here. Okay, all of you, any doubt? Sure. So now ligand based drug designing is on keys method. That means you are just deriving the information about by itself. It also has some ligand. You can take out that ligand and you can study its structure also. Okay, so when the XRD studies gone into detail, then we came to the conclusion, or we get the library of protein. जब XRD डेवलप हुआ 1980 के बाद ये सारे डेवलपमेंट हुए, उसके पहले नहीं. Okay, earlier we were with lock and key only. Okay, so fix, okay, both, and we are making it mix, right? So now next points थे जो हमको face करना पड़ रहा था, right? So protein structure has a large degree of flexibility. Of course, एक protein का structure, अभी now you have studied, I think, primary structure. You have studied secondary structure, you have studied tertiary structure, you have studied beta pleated structure. So it's a big deal. Okay, so कितना complicated है now the things. So then the CADD is called as pharmacore approach. Okay, so now this is QSAR, that is called as quantitative structure activity relationship. ये इसके साथ कैसा मैच जाएगा, बस ये देखना है. That is given by QSAR. QSAR, suppose, say for example, this is the receptor site and there is the active site, and in this active site, if suppose this hydrogen is there, due to the change in the polarity of the receptor and the ligand, there will occur the hydrogen bond. That is called as H bond. That H bond is must. Otherwise, ligand will not interact with receptor. So now, if suppose this is a good thing, so you call it as a pose. आप मेरी फोटो खींचो, मैं ये पोज देती हूँ, correct? So that is called as pose. So this ligand fellow is giving the pose. So like this, you can have 500 thousands of poses, which you can make it on computer. But still we have some limitations in that. हम लोग 500 तक ही कर पा रहे, thousands तक ही कर पा रहे, but nature में बहुत ज्यादा है. Are you getting my point? Pose is important. Conformer, एक तो मैंने यही बताया आपको. So structure like this, space में arrangement, okay, cis trans पढ़ा है आपने, okay. So all that you can connect here. So now here there are two possibilities here. One is called as known protein structure, SBDD, okay. Now these two together is called as computer aided drug design, okay. Now here in SBDD, protein ligand docking, no known ligand is there, suppose, then it is called as de novo. I just had a talk with you, de novo, startup, okay. New molecules, everything new, I want to start with new, okay. So and actually in de novo also you cannot start randomly. What you have to do, you have to study the library of the known ligands, okay. फिर जो एक्सपीरियंस जो लोग होते हैं, देन दे ट्राई टू कम अबाउट कि हाँ, अगर ये वाला मॉलिक्यूल कोर मॉलिक्यूल लिया जाए और यहाँ पे ये दो स्ट्रक्चर चेंज किए जाए तो शायद वो एक्टिव बन सकता है. Are you getting my point? It comes by experience, or in means if you will go with the drug design, then you will get into that. So that is called as totally a de novo. De novo, you start with the new, okay. That means literally means start with the new. Now protein ligand docking, it is either ligand based, okay. You can have one or more ligand, okay. And similarity in searching, there may be several ligands, possibility is there. And pharmacology approach says you can start with, you can have 20, only one receptor is there, only one cancerous protein is there, try 20 ligands, no problem. एक की 20 चार, it is as good as that, okay. So this comes under ligand based drug design. And this is not only 20, we are going like anything. So CADD is of no use, it needs experimental data, okay, and some sort of. अब ये experiment de novo है ना, तो स्टार्टअप को कोई जल्दी से ये नहीं मिलता है, राइट. स्टार्टअप को थोड़ा जद्दोजहद करनी पड़ती है, राइट. So we are into that, yes. So now this is called as virtual screening. Now we have to put a filter, okay. Now virtual screening is the computational or in silico analog of biological screening. Biological means the way you, okay, the way they will behave, you can see the same interaction here also. Where in computers will tell that whether the hydrogen bond will be there or not. If it is there, then what will be the energy between them? So all these things are just calculated theoretically, okay. So now see, suppose I was told to make 50 molecules in my lab, okay, and I was told to allow it for clinical trials. First you go for this one, in vitro culture, then I'll go to microbiology department, then they will do the experiment, then they will give the result, then I'll go to the main that is cancerous protein or say another proteins, and then if I'll do that, then the process is going to go a very, very long process. Okay? Then after getting from there also positive, then they will go for, I think in COVID, you might be knowing that there were this much number of candidates who were there for available for vaccine. First 300 was the taken as a sample, then 300 was changed into the longer view, like that. धीरे धीरे उसका sample size बढ़ाया जाएगा. So clinical trials is first done in animals like rabbits, rats, and everything, and then it is being going to the human beings. those who are ready, and then you will get to the come to the drugs. Okay, that too, it has to undergo a lot of approval processes. Got it? So it takes 14 years, right? It is not of matter of 2 to 4 years. Now, how the time is reduced, this computational study will tell you whether hydrogen bond will be there or not, whether the interaction will be there or not. अरे नहीं होता है तो छोड़ देना यार, क्यों ले रहे हो उसको, काहे दिमाग लगा रहे हो, दूसरा ले लो, मॉलिक्यूल दूसरा ले लो, राइट? सो आपको फिल्टर करने में मदद होती है. Are you getting my point? That is screening, right? And is to score. ठीक है, चलो इसको स्कोर लेते हैं. अब स्कोर यानी क्या होता है? स्कोर को मैं थोड़ा सा डिटेल में डील करूंगी. Rank and filter a set of structures using one or more computational processes. So there are more. It can be, now how does it help to screen the compounds experimentally? Libraries खुद का बना सकते हैं, right? Which compounds to purchase from external company, that is important to analyze the results of the experiment, okay, such as HTS run. Now these are all the filters, okay? So आपको मिल क्या रहा है? Structural filters, drug like filters, ADMET properties. अब इसमें से अगर actives known हो गए तो ligand base से जाइए. अगर आपको 3D structure का target पता चल गया तो you go with structure based method. Structure based method is again a protein ligand docking. This is again a ligand based method. Ligand based method that comes under cheminformatics. जैसा आपने सुना बायो इन्फॉर्मेटिक्स, वैसा एक और इन्फॉर्मेटिक्स है, दैट इज केमी इन्फॉर्मेटिक्स. केमी इन्फॉर्मेटिक्स डील्स विद द ligands only. They want to know the positions, they want to know the score, they want to know the I should say binding in interaction between the two fellows, the proteins as well as ligand. Got it? So there comes here pharmacore based drug based design, that's similarity, and then comes the बायोलॉजिकल कंपाउंड एक्विजिशन. अब आते हैं हम लोग एक्विजिशन पे. ओके, सो ये इतना इजी नहीं है, इट इज अ वेरी बिग डील. अब इसमें दैट इज कॉल्ड, अब फार्माकोफोर क्या है? अ सेट ऑफ ज्योमेट्रिकल कॉन्स्टेंट्स. अब ये आपको एक पर्टिकुलर साइट है, ये इस टाइप का साइट चाहिए, राइट? सो ये इसके साथ सिमिलर है, ठीक है? ये लाइव एग्जांपल है. ओके, ग्लेंस फार्माकोफोर. ओके, ये फार्माकोफोर. अब इसमें यहाँ पे जो लिगैंड्स है, ये दैट इज द स्ट्रक्चर ऑफ मिथरसिजाइड एंड मार्क्ड विद फार्माकोफोरिक एलिमेंट्स. ओके, सो दिस साइट you can see hydrogen bonding, HBD, hydrogen bonding donor, a hydrogen bonding donor. About this, now overview is what? Activity data will generate the pharmacophore. Pharmacophore मतलब एक region, एक active site. So region, that pharmacophore will be created. You will be getting the target. This is what is the target, that is colored with blue. And then you are seeing the different in the libraries. You are allowing it to hit with it, the receptor, and then you will get the perfect thing, and you can buy those hits. And test this. No need of buying. This is not required in such some pharmacophore approaches. So now in your syllabus, more stress is given on this computer-based drug design. So I'll go into the detail of that. Pharmacophore में तकलीफें है, okay? So that means उसका पहले identify करो कैसा होता है. Hydrogen bond donor, acceptor, lipophilic groups और charges. ये सबका एडिशन होगा वहां पे, और उसका टोटल एनर्जी काउंट विल बी देयर. ओके, और उसका काउंट होने के बाद दैट यू विल गो फॉर द स्कोर, और स्कोर होने के बाद रैंकिंग होगी, विच इज द बेस्ट. ठीक है, समझ में आ रहा है क्या बोल रही मैं? इट इज एज गुड एज. अभी मुझे बोला जाए कि भाई ये 10 चीजें हैं, मुझे बताओ इसमें से बेस्ट कौन सा है. सो मैं 10 को तो बेस्ट नहीं बोल सकती हूँ. सो आई विल, मेरा कुछ स्ट्रैटेजी होगा, तो मैं वो स्ट्रैटेजी से उसको स्कोर करूंगी. स्कोर करने के बाद मैं उसको, ठीक है, रैंक करूंगी, राइट? तो अब आपको रिलेट हो रहा है स्कोर वर्ड, ओके, रैंक वर्ड. सो ये 2 वर्ड्स जो है वो बार बार आएंगे. ओके, सो दैट यू हैव टू गो. दिस इज व्हाट इज द प्रोटीन. प्रोटीन के अंदर जो साइट है दैट इज कॉल्ड एज बाइंडिंग साइट. In that binding site, the ligand is fitted, lock and key, as good as that. But this, now see, this site is flexible. So then the thing will go wrong. So you have to go for multiple ligands. Are you getting my point? So multiple ligands means now the chances are there. Okay, naturally, if I know this ligand, अच्छा यहाँ पे ज्यादा जगह है क्या, चल ये वाला ग्रुप डाल के देखते हैं क्या होता है, चलो ये वाला ग्रुप डाल के देखते, like that. So you can do the changes in ligand. Second thing possible, इसको change करके देखो. Okay, so if this can be altered, but this is the natural thing which is coming out, but still we are trying कि इसको change करने से भी कुछ होता है, like that. So that will come, this will come as receptor based drug design, and this will come under ligand based drug design. Okay, got it? समझ में आ गया सबको? कहीं एक चीज कांस्टेंट है, एक चीज फ्लेक्सिबल है, कहीं दूसरी चीज कांस्टेंट है, पहली चीज फ्लेक्सिबल है. गॉट इट? अब ये कॉम्प्लेक्स बन गया, ड्रग अंदर एंट्री ले लिया, ठीक है, मना कर दिया, काम नहीं करने दूंगा, सेल को बोल दिया भैया तेरी नहीं चलेगी अब, ठीक है, राइट? तो अब उसको सिग्नल देने से ये रोकेगा, राइट? इस तरीके से ये आगे जाता है, राइट? So this is structure based drug designing method. Okay, you are using a protein structures and you are producing the mimics. अभी इसमें एक वर्ड है mimics. Mimics means उसके ही जैसा. Duplicate सुना? Mimics. So now it predicts the pose. Pose दिखाया, ये मॉलिक्यूल ने अपनी पोज दिखा दिया, ठीक है, उस पोज पे वो जाके फिट बैठ गया, राइट? अब फिट बैठने के बाद आपको एक बाइंडिंग एफिनिटी मिली. एफिनिटी इज जस्ट द एडिशन ऑफ एक्सेप्टर, डोनर. अभी हमने बहुत सारे चीजें गिराया आपको, ओके, दैट इज लिप चार्जेस. ओके, सो दोस एडिशन इज कॉल्ड एज स्कोर. अब स्कोर से आपको पता चला कि उसमें स्ट्रेंथ कितना है, तो आपको करना क्या है, एक एल्गोरिदम सेट किया है, उस एल्गोरिदम में से स्कोरिंग फंक्शन निकालना है, कितना होता है, एंड देन अगर उसमें डिफिकल्टी है तो किस पार्ट में डिफिकल्टी है. अभी मैंने आपको बताया भाई ये एच नहीं जम रहा है, तो किस पार्ट में डिफिकल्टी है, स्ट्रक्चर, लाइगैंड स्ट्रक्चर. चेंज द लाइगैंड. रिसेप्टर में अगर डिफिकल्टी है, यू कैन जस्ट चेंज द फ्रैक्शन ऑफ द फ्रैगमेंट. समझ रहे हो क्या बोलना चाह रही हूं मैं? उसका एक्सआरडी जब आप लेंगे, जब आपका उसका एक्सआर स्टडीज आप स्टडी करेंगे प्रोटीन का, प्रोटीन इज आल्सो ए फ्लेक्सिबल. ओके, सो उसका एक अपना सॉफ्टवेयर है, और वो सॉफ्टवेयर मैं आपको बताती हूँ. प्रोटीन डॉकिंग स्टडीज भी होती है, और प्रोटीन लाइगैंड डॉकिंग स्टडीज भी होती है. ओके, सो वैसे दोनों सॉफ्टवेयर्स है, जो आई शुड नॉट से दोनों सॉफ्टवेयर्स है, दो ग्रुप ऑफ सॉफ्टवेयर है जो आपको दो अलग अलग बातें बताते हैं. प्रोटीन अगर इंटरेक्शन देखना है तो यू कैन गो फॉर ग्लाइड, ठीक है, ऐसे सॉफ्टवेयर है ग्लाइड. एंड देन इफ यू वांट टू गो फॉर ligand based drug designing, okay, then you will have Argus Lab, Autodoc. Okay, these are the two free softwares which are there present in the internet. Okay, the others you have to purchase, right? So they are licensed software, so you have to purchase them. But these two are very easy to operate also, and tutorials are also given. So now what does this algorithm will do? They will generate the poses. Okay, pose बताएगा आपको 500 में से कौन सी फिट है, सही जाएगी. सेकेंड आपको ओरिएंटेशन बताया, सिस, ट्रांस, कन्फॉर्मस, एवरीथिंग, और फिर उसके पर्टिकुलर कन्फॉर्मेशंस में से क्या जाएगा. सो यू कैन सी दिस. यस, सो चेयर एंड बोट कन्फॉर्मेशंस, राइट? सो नाउ दिस इज व्हाट इज कॉल्ड एज ड्रग टैक्सोल. Okay, now Taxol is that's an anti-cancer drug. Okay, and you are obtaining this drug from which tree? पैक्लीटैक्सेल सुना है? यूटी नहीं, इसका ढूंढो, नाम ढूंढो. Correct. Okay. So this, what we are seeing here, you can see the bond here. Okay. Now, when you have done the in-depth study, if oxygen ring is present in the structure and this type of side chain is there, the molecule is more active with the binding site receptor of cancer. Understood or not? So what I'm doing, I'm just finding out the structure carefully. Got it? So here what we are doing now, this we are getting the different conformations. स्ट्रक्चर को घुमाते चलो, ठीक है, पूरा मॉलिक्यूल घूमते जाएगा, राइट, एंड यू विल हैव द डिफरेंट conformers, right? So you will have this many number of conformers, and then you will get this rotatable bonds and combination explosion of the study will be there. Okay, so रिंग कन्फर्मेशन भी बदल सकती है, रिंग की पोजीशन भी तो बदल सकती है ना. ये एक इस टाइप की रिंग है, यही नीचे कर दिया, यही ये नीचे, ये दोनों ऊपर है, इसी को मैंने नीचे कर दिया, ऐसा फ्लिप कर दिया, तो इट विल चेंज टू चेयर फॉर्म. ये आपने पढ़ा है, राइट? सो ये जो है, अब हम लोग इसमें आते हैं रिजिड डॉकिंग. मैंने आपको बताया ताला चाबी, दैट इज अ रिजिड डॉकिंग. रिजिड डॉकिंग पहले होता था, राइट, but after that the ligand was treated as rigid structure during docking, and only these energies were given the freedom कि भाई मॉलिक्यूल ऐसे ही मूव कर सकता है, इससे ज्यादा नहीं कर सकता, right? So there when freedom was not given to the ligand. Okay, but slowly what had happened now, flexible docking has come. So de novo, जब इतनी बड़ी बात हो रही है, startup. Okay, you have the library of 500 molecules at a time, so you can have this. So I told you, no, you can have, if you want, you can have the photo of this. So the common approach to use is stochastic approach search methods, quite difficult. Okay, so because everything is, if will go flexible ना, मी तुम्हाला तुमच्या हातात कणिक दिली, एकीकडे एक कणिक आहे आणि दुसरं मी तुम्हाला रेड कलर टाकून दुसरी कणिक दिली आणि मी तुम्हाला सांगितलं चला डॉकिंग करूया, म्हणून दाखवा मला. व्हाट विल हैपन टू द प्रोडक्ट? कुछ मिलेगा क्या? बनेगा कुछ प्रोडक्ट? बोथ आर फ्लेक्सिबल, चाहिए वैसा घुमाओ, सब घूम रहा है, ये भी घूम रहा है, वो भी घूम रहा है. ओके, सो दिस इज नॉट पॉसिबल, राइट? सो दैट्स व्हाई वी आर इनटू कि भी कौन सा पोज सही है, जो पोज सही है उससे लेके आगे बढ़ा जाए. आर यू गेटिंग माय पॉइंट? इसलिए मैंने आपको बोला कि पहले एक रिजिड्स का स्टडी कर लेना चाहिए, देन यू गो फॉर फ्लेक्सिबल. ओके, सो नाउ this is flexible docking is more now handling the protein conformations. So rigid receptor approximation is there. अगर इसको receptor को अगर rigid किया तो फिर ये part आपको मैं बता चुकी हूँ अभी. Okay, verbally I took all the things, right? So larger protein movements can be only handled by separate docking with different protein conformations. Okay, so you take different proteins, not only cancerous protein, you take any other protein. There's a PDB database, complete database with PDB files, and they are having a lot many, I should say, deceased protein, and then those deceased protein you take your choice and then take the help of that disease and you will get into that. Okay, so अब ये क्या मिलेगा आपको? You will get the binding affinity. Okay, now so the score should be the score of the correct pose will be active, higher. ठीक है, then what he says, अब ये part जो है force field based, empirical based और knowledge based, ये type के जो ये gray color से लिखा है, these are all softwares. Okay, so these, okay, in my research I have used Autodoc. We can go for ChemScore Glide. Now again, this is what protein. Again, it will give you the affinity. If it is knowledge based, then drug score and ASP, these things, right? So I hope you understood this part. Okay, may I move forward? बहुत ज्यादा तो नहीं हो गया ना? डाइजेस्ट हो रहा है? ओके, डाइजेस्ट हो रहा है क्या? हाँ, yes.
Earlier mentioned that to generate a pharmacophore, protein structures are not required. So what is
Protein structure are not required is not there. We are taking the protein, we are finding out the active sites and generating a pharmacophore like that. A mimic, we are generating a mimic and then we are going into that. Okay, actually you have to deal with that, there is no choice. But these softwares are providing that protein also, guide software. Even I have not studied much about it, but it is showing you protein protein interaction. You have to take the fragments and then you have to go for it. So now see, you have to, now this is what I wanted to go into detail, that is delta G value. Okay, now what is delta G value? Change in free energy. All of you please write down one formula. Delta G is equal to, you are 12th passed, so you can write that formula please. Delta G is equal to delta H minus T delta S. ठीक है, अब ये nothing is moved at all and the things are remaining in the still condition, that means it is ordered state. If the things are here and there, the things are disordered state. So more is the disorderness, more the system is stable. More is the disorder. अरे बच्चे ने अगर ठीक से पढ़ाई किया है तो उसका टेबल में किताबें इधर उधर रहेगी. मुझे देखना है, मेरे बेटे को पकड़ना है, मैं बराबर पकड़ लूं. कैसे? उसके टेबल पे किताबें देखकर. Right? So whether the work is done or not, can you study like that? Yes. So delta S value, chali, you make it as more. Delta G is equal to delta H minus T delta S. Let us consider delta H as 5 and delta S as 5. ठीक है? Let the T be constant. Temperature को constant रख. ठीक है? अब बताइए, suppose if I increase the value of delta S from 5 to 10, what will be the delta G value? It will be towards more negative. So more is the negative delta G, more is the spontaneity of the reaction. You have studied in 12th standard. Okay? If delta G is equal to negative, then you say that the system is spontaneous. If delta G is 0, then it is equilibrium. And if it is positive, then it is non-spontaneous. No receptor, no binding, no affinity. So now what I want from this algorithm? Either you talk in that way, or better he is telling you the this one, that is the delta G value. G binding energy should be, are you getting my point or not? I'm more in will be acceptable. So more negative is the delta G, more is the free energy of the system. Got it? And the more successful, more score, more good score, more ranking will be there. Now what is delta G now here? It is binding of all the things. These things will be there. Okay. So what you are doing accurately, whether you are getting the correct pose it or not, if you are getting 70, you can predict the pose 70. Right? So ये computer ने आपको बता दिया कि just shortly there are some steps in the tutorial you have to go. So okay, but right now to cut in short, I will say that sometimes already water molecules are there. You remove that water molecules, you have to add hydrogen to that in that structure, and then you have to carry out the dock like that. So then you have to create the active site, then you have to create the grid, then you have to attack with the molecule, and then the molecule attacks, and then you get the delta G values, that is in the form of score. Got it? You have to calculate HBA, HBD, like that. Everything you have to calculate there in that structure, and then you do the docking. And when you run the docking, you will get the score. More negative score, more good is the result. Got it? Perfect. So this is what is the structure, and that is called about the drug designing. This is all like, you know all these things, right? More better than me. Okay. So this is all about our drug designing. Drug designing is a software. Okay. Now I want you to I want to know what projects you are doing so that I can explain you You are doing with some projects. You can ask me. Yes, please. Anybody? Yes, Shraddha. Any questions? You are doing some project? Okay. Anybody doing on bioinformatics? You can raise your hand, please. Yes. Yes. What study is going on? Can you please tell me? So you are basically dealing with the receptor. You are just taking the protein into consideration. What is it then? Amino acid sequence is what? Protein, na? You're working with protein, right? Please be seated. I hope you can connect now. Okay, so you're working with protein, right? Yes. Anybody else? What you're doing? Same project. So you have done the sequencing. You have separated the fragments. Correct? Am I right? Now you will go for next step. Will be this. Understood or not? Okay. Yes. Anybody else is doing on bioinformatics? No one? There are many other good topics. Okay. See, one more challenge I'm giving you. We are from Ayurveda. Okay. Now, Indians are said to be very good at Ayurveda, right? So, ask your Gurujis, ask your all Dadajis and all, and just come to some molecules which are very good for certain type of disease. Take out that plant, find out the phytochemicals which are present in it, okay? Change that ligand with some molecules by seeing the present drug. Are you getting my point? You see the first present drug. What is that drug? Okay, the drug is this. It consists of chlorine. So, okay, you can just introduce that chlorine to that uh, structure obtained by Dadajis. Okay, so that plant, okay, that is called as phytochemical. So we will what we will do? We will take synthetic, go to natural, make it semi-synthetic, and then induce in the for the drug design. So now, of course, it will not have side effects. Am I right or not? So I believe not in Ayurveda, I believe not in allopathy, but I believe that the things which will go well. Are you getting my point? Without getting any of the side effects. That is what my target is. I hope you are getting my point. Okay. So that's why I want to relate the natural thing, artificial things, and the things together and get it the better way. Got it? If I get the pizza out of maida, why not with millets? Why not with jawari? Why not with bajra? Are you getting my point? If suppose this thing comes into picture, Domino's will not survive here. Correct? So let's go in that way. Let's go with de novo and then design your own molecules, have 500 compounds library, get a good score, get a good ranking, and then have some empirical values, good values, and then go for drug design. Understood this? Pakki baat? Last part I want to tell you. I have heard from the president of Dr. Reddy's lab that I want to share with you. That is called as green chemistry. He was talking on green chemistry basically. What he said, suppose you are making one synthesis, दवाई बनानी है, तो दवाई बनाने के लिए A से B, B से C, C से D, D से ऐसा करके मैं H तक जाती हूँ मेरी दवाई बनती है, राइट? मुझे ए से एच तक जाना होता है. बट व्हाट ही सेज, go for atom economy. What is atom economy? तेवढं घ्या जेवढं लागते. समजलं का? जास्तीचं घेतलं तर साइड इफेक्ट्स होतात. Are you getting my point or not? So try the starting structure. Go from A to B, that's all. At the most C, not with H. Are you getting my point? Okay. So side products, side chains, all these are very difficult, right? So then things become in a very wrong way and we get into that. Got it? All of you? Pakki baat? Okay. So next time I want more students from bioinformatics to have with more Ayurveda phytochemicals mixed with some new knowledge and get into with new structures with good delta G values. Good means how much? More negative delta G values, right? Yes. So all the best, okay? And let's conclude the session. Yes. Any questions if you have, you can ask. Yeah. Thank you so much.